I'm Dr. Sarah Best, and I co-lead the Brain Cancer Research Laboratory with Dr. Saskia Freytag, bioinformatician, and Dr. Jim Whittle, a neuro-oncologist. And together we're interested in understanding the underlying heterogeneity of brain cancer. Brain cancer is a devastating disease with a 5-year survival rate of just 22%, well below the national average and that of other cancers. Brain cancer can be subdivided into a number of different subtypes, with the gliomas having the poorest prognosis, and glioblastoma with just a 5% 5-year survival rate having by far the highest diagnosis in Australia. Gliomas arise from glial cells in the brain, such as astrocytes that transform into astrocytoma and oligodendrocytes transforming into oligodendroglioma. These tumours further transform into high-grade gliomas, anaplastic astrocytoma and anaplastic oligodendroglioma respectively. The other form of high-grade glioma is glioblastoma or WHO grade 4 glioma, and this is a devastating disease. These tumours can be characterised by a mutation in IDH1, where WHO grade 2 and grade 3 gliomas typically carry the IDH1 mutation, whereas glioblastomas do not. And they can be further characterized by chromosomal gains and losses, such as gain in chromosome 7 and loss of 10 in glioblastoma, and loss of 1p19q in anaplastic oligodendroglioma. These glioma cells can be subdivided into 5 different cell states, and this is based on the fetal brain, where these brain cells could be subdivided using single-cell RNA sequencing into progenitor and differentiated oligodendrocytic, astrocytic, mesenchymal, and neuronal cell states. And these cell states hold true when we look at glioblastoma as well, where the majority of the different cells in glioma fell into these 5 different cell state categories. Tumours display varying levels of heterogeneity based on these different cell states, such as mesenchymal enriched tumours or tumours that are enriched in oligodendrocytic cell state, but we also see a high number of mixed cell state tumours. And so in the lab, we're really interested in understanding the spatial distribution and organisation of these different cell states in patient gliomas. So for this project, we took high-grade gliomas, our IDH wild-type glioblastomas, and IDH mutant anaplastic astrocytomas. And both of these tumour cell types have high expression of the marker GFAP in their tumour cells, and a similar proliferative index when we look at proliferating cells by Ki67. For our program, we selected 3 anaplastic astrocytoma samples and 3 glioblastoma samples, and we investigated the differences in spatial heterogeneity between these. So to perform this, we used the NanoString geomics platform, and this allowed us to take our FFPE archival tumour samples and stain these using GFAP for our tumour cells, Ki67 for our proliferating cells, and CD45 marking our immune cells. We selected regions of interest based on the pathology of the tumours, and we're able to isolate different areas of illumination in each region of interest for whole transcriptome analysis. It was first important for us to look at the pathology of the tumours that we had selected and to identify regions of interest for our staining. So these are our 3 anaplastic astrocytomas and glioblastoma patient samples. And we selected 5 to 7 regions of interest within each tumour. And using our pathological classification of these, the majority of these different regions of interest were in our cellular tumour portions, but we also selected regions such as this one here in AA3, where we have infiltrating tumour and leading edge, where we can look at the cellular tumour out through to normal brain. Using the CD45 immunostaining, we were able to determine whether each region of interest had high or low immune infiltration. And using our Ki67 staining, in addition to the pathology, we could assign tumor or border to each section, in addition to Ki67 negative border, because we had significant regions of tumor that had high proliferation, bordering regions of tumor that had very low proliferation. And in green, we also had regions of normal brain. So from our studies, our 3 anaplastic astrocytomas, or AA, and 3 GBM patients, for our areas of illumination, we had 16 AA tumor regions versus 18 glioblastoma regions. We had 5 regions of interest with Ki67 positive areas of illumination for both AA and GBM. And for our immune infiltration, our CD45 areas of illumination were 15 for GBM and just 5 for AA. First, when we deconvoluted the different cell states based on the 5 cell states I introduced earlier, we could identify in our 6 tumor samples that GBMs have higher regions of progenitor and mesenchymal cells, whereas our anaplastic astrocytomas have a significantly increased proportion of our oligodendrocytic cells, really highlighting the differences in the classification between AA and GBM. When we split these up between our different regions of interest, we were really interested to see that there was increased heterogeneity across the different regions in our GBM samples compared to our AA samples that retained a similar portion of each cell state no matter where we were in the tumor. And if we look a bit closer into our GBM samples in GBM3 region 4 and 5, we can see that these 2 regions that are right next door to each other have the emergence of an entire population of astrocytic cells, whereas in region 4 we have a complete absence of this population. So we're really interested to identify these very significant differences in spatial heterogeneity of these different cell states within the same piece of tumor. Because we investigated our proliferating cells using Ki67, we were able to split our areas of illumination of our tumor into proliferating and non-proliferating. And we identified that, especially in our glioblastoma samples, we see significant enrichment of the progenitor cell state in our proliferating GBM cells compared to our non-proliferating cells. And this is a very interesting finding, and it also validates previous work which highlights the progenitor cell state as a more cycling cell state in GBM. Next, we use the transcriptome data to identify the copy number alterations, and this was using CNV-KIT, which predicts copy number alterations based on clusters of transcript gains or losses relative to the immune compartment, which we know should not have any cancerous mutations. In this region of GBM1, we have a region from ROI4 where we have our cellular tumor through to ROI6, which is our normal brain. And we were able to use the copy number alterations to identify and validate the normal brain compartment. And so in this chromosomal array, we have our different chromosomes in each column, and each row are our different regions of interest just in GBM1. And we can see in our row 1 at the top, this is our region of interest 6, which is our normal glia. And we can see the absence of these clear copy number alterations, such as loss in chromosomes 11, 13, and 15. And so this really does confirm to us that this region does contain normal glial cells. Next, when we looked at the anaplastic astrocytoma samples using the copy number alteration, we identified anaplastic astrocytoma sample 1 as having very clear 1p19q chromosome arm loss. This was consistent with a significant increase in the oligodendrocytic cell state across all of the different tumor regions of AA1. And when we look at Shannon entropy scores across the AA samples, we can identify here these oligodendrocytic cell states, no matter in the proliferating or non-proliferating compartment, are really driving differences between the tumors in the AA samples. And so this suggested to us that perhaps this isn't an anaplastic astrocytoma and may in fact be an oligodendroglioma. So we went back to the clinic to identify if there'd been any updates in the diagnosis of this patient. And we found that in the interim, the patient had been re-diagnosed to anaplastic oligodendroglioma, and this was based on an independent whole genome transcriptome study, which identified the 1p19q loss. And so this further validates the effectiveness of our copy number alteration analysis using the geomics data. Next, we wanted to really drill down on that spatial heterogeneity in regions of the tumor that are very co-located. And this example is in GBM1, where we can see regions of interest 1, 2, and 3 that all had high immune infiltration. They're all spatially co-located in the tumor. And we could use the copy number alterations to further investigate the different clonality that these tumors arose from. And this really strongly showed us that tumor region 1 had very different copy number alterations compared to tumor region 2 and 3, which had very similar alterations between all of the different areas of illumination. And this includes loss in chromosome 10 that's present in tumor regions 2 and 3 and not in tumor region 1. And so this really reveals a different clonal evolution of the tumor using this geomics whole transcriptome data. So next, we could use the sum of squares approach to really investigate intra and intertumoral heterogeneity. So really looking at the differences between the different tumors in each cohort and also within the different regions of interest within those tumors. And this schematic just shows us how we can use the sum of squares to really investigate the differences within. In this example, there's small differences within, whereas this tumor has very large differences, and then the differences between, so the distance of each of these histograms to the center. So in this example, I have shown the 3 GBM samples, and we're looking at the heterogeneity between and within the tumors in these. And I've just highlighted on this plot some key genes or chromosomal regions where we see differences with the intra and intertumoral heterogeneity. But as an example, I've selected PDGFRA, and we identified this as being significantly heterogeneous between samples. When we looked at the transcript expression of PDGFRA, we could see that GBM2 indeed have very high transcript of PDGFRA compared to GBM1 and 3. And this was consistent with very high protein expression in this tumor. So this just reveals that we were really able to investigate the intratumoral heterogeneity using this sum of squares approach from the CNV kit data. Next, we deconvoluted the immune compartment using the CD45 positive areas of illumination. And before I go into that, this is protein data looking at the amount of immune infiltration across each of the 3 tumors. And we can see that there were variable degrees of immune infiltration with much higher infiltration in GBM2 and 3. And when we look at CD68, which marks for our macrophages and microglial cells, we can actually see that we have much higher proportions of macrophages and microglia in GBM1 and 2 compared to GBM3. So this difference in immune infiltration in GBM3 is not relative to the myeloid population. So we can actually assume a lot of lymphoid infiltrate. But we could deconvolute the areas of illumination to identify which cell types were present in these different regions of interest. And similar to our protein analysis, we identified that GBM1 and 2 had much higher immune infiltrate of macrophages and microglia in these two blue shades here, whereas GBM3 had much higher proportion of lymphoid infiltrate in our orange and red bars and a lot of neutrophil inflammation. And so this was consistent with what we saw in the histology analysis. When we broke down the immune infiltration by the different regions of interest and also compared this with the different cell states within each of these regions, we can actually start to identify some correlations between different tumor cell states and different immune infiltration. And here I highlight when the regions of interest had high astrocytic cell state in this blue region of the bar here, we see very high levels of microglia and macrophage inflammation. And in these samples on the left, where we see higher mesenchymal cell states, we see very high levels of lymphoid inflammation and also neutrophils. So we do start to see some very specific associations between different cell states and immune infiltration. So we could quantify that by looking at a correlation plot. And in this plot, each column and each row highlights all of the different tumor and immune cell types that we've identified. And the value shows us the correlation between these. And the more yellow the values, the stronger the correlation. And we're first able to identify from this correlation plot that we have three different niches within our tumors. We have our strong tumor niche, which is characterized by our astrocytic, microglial, and macrophage cells, which are highly represented in this niche. Our outer niche, which contains a high number of oligodendrocytic cells and lymphoid cells, and our mesenchymal niche, which contains a high number of monocytes and neural progenitor cells. So we found these different niches in our genomics data, but this was just 3 patients. So we went out to the literature and identified a cohort of Visium data that was performed on up to 18 glioblastoma samples. And here I'm showing you one representative example of this. This is a H&E from the Visium data, and I've just outlined the regions of tumor in black, a mesenchymal patch in orange, and an outer patch in green. And this correlates with our 3 niches that we identified in our correlation plot. So when we look at the different cell states that are present in each of these regions, we see, consistent with our data, that we have a high number of astrocytic cells that are marked in red here. In our outside niche, there are a large number of our oligodendrocytic and neural cells. And in our mesenchymal patch, we see a very high and co-localized proportion of our mesenchymal cell state. So to give one example of a very high correlation we identified, this was between our mesenchymal patch and our monocyte cells. So we investigated the Visium data to have a look at a monocyte cell signature and to correlate this with the different niches that we'd identified. And consistent with our genomics data, we also found in the Visium that our monocyte score was significantly increased in the mesenchymal patches, really identifying to us that these 3 different niches and correlations between immune and tumor cell states are significant and can be more widely identified. So this is a schematic of the different co-localization between the different immune and tumor cell states, where we can see within the tumors we have a high number of our astrocytic and microglial macrophage cells, whereas out in the outside of the tumor we see more of these monocyte progenitor mesenchymal patches. So next we wanted to better understand the conversations that are happening between these cells and why they're co-localized. And to do this, we needed to do receptor-ligand interactions. And here I'm showing an example plot where we looked at a large number of receptor ligand interactions and we're just interested in those that created particular patterns when we compare all of the 4 different combinations of tumor and immune. And so we were interested in ligands and receptors that were talking from the tumor cells to the immune cells to better understand these niches. And so this is a short list of those different ligand receptor pairs that were really specific to the tumor immune interaction. And when we look back at the association between astrocytes and our macrophages and microglia, now we can identify which ligand receptor pairs are key in that interaction, where we see this MIF, CXCR4, and HMGB1 HAVCAR2 relationships that could really be driving these key interactions and co-localization. We also identify in our mesenchymal rich regions where we saw a lot of lymphocyte inflammation and neutrophils that there's a high TGF-beta signaling pathway that's activated in between these different cell states, which does explain a high level of inflammation. And so this was really useful to us to better understand the niches and the different co-localizations in our data. But to better understand the heterogeneity and the different cell states at a single cell level, we next wanted to turn to the CosMx data and better understand at a single molecular level what is the expression of our different genes. And so to do this, we use the CosMx TAP program, just looking now at GBM1. So we submitted the same FPE block, and this was co-stained with DAPI, GFAP, histone H3, and 18S rRNA. And then the 1000 probe panel was applied, and we were able to use the cyclic in situ hybridization to amplify and identify these different probes in our tissue. So the probe panel that we used was the human immuno-oncology panel. And I'm just showing here an examination of the probes present in that panel and how we could use them to identify our receptor ligands, our tumor cells, and our immune cells. And so this upset plot just shows us how many probes we were able to use from this panel, and that we had a very high number of probes in the immune panel that allowed us to deconvolute our different tumor cell states that I showed previously. So we knew before we performed the CosMx analysis that we would be able to identify our different cell states. So this is an overview of the experiment that we performed. And so on the left, we have our geomics regions of interest in these black boxes. And I've just outlined the region that was investigated by CosMx analysis. And if we flip that over, we can have a look at where the fields of view were identified by CosMx. And we investigated 7 fields of view in this region of tumor that we'd previously analyzed by geomics. And we were able to identify a high number of cells and a very robust mean transcript per cell. Because this data is essentially single-cell transcriptomics, we could use all of our favorite single-cell tools on the CosMx data. And so this just highlights that from GBM1, with our 7 fields of view, we identified 11,801 cells, and this clustered into about 35 different independent clusters. To better identify the major cell types from this data, we were able to use these 3 key genes, as well as a number of others, to ascribe the immune population, which we can see quite highly expresses CD163 and is at the top of the UMAP here. ApoJ was highly expressed in our tumor cells, and COL4A2 was highly expressed in our vasculature. So now we can really delineate these key different cell types. And then applying our cell state deconvolution across to these cells in the UMAP, Now we see our mesenchymal cells, our astrocytic cells, progenitor cells, and our microglia and macrophages as well. So we were really able to robustly identify all of our different cell states in the COSMIC data. And to further dive in to the heterogeneity, the spatial heterogeneity that we could find, an example here is FOV 9, 10, and 11, where in FOV 9 we see a high number of mesenchymal cells in a patch together, but right next door in FOV 10 we see a very strong astrocytic and progenitor patch. And this is carried across into FOV 11, where we see these astrocytes and progenitor cells and some vasculature in this region as well. And so this example just really highlights just how heterogeneous these different cell states are in very small regions of view. And we really need to better understand these different niches and how the cell states and immune cells interact within each of these niches to better understand how we can use this information to improve treatment for glioma. And so I'll leave it there and like to thank the amazing team that helped with this work, and especially Dr. Saskia Freytag, an expert bioinformatician who has really led all of the analysis of this data. Saskia will be live at the end of this call to be able to field any questions you have. I'd also like to acknowledge Joel Moffat, who also drove a lot of the bioinformatics analysis, and our anatomical pathologist, Oluwasegun, and the amazing lab that we have full of wet and dry biologists to better investigate these very complex questions in glioma. I'd also like to acknowledge at the Royal Melbourne Hospital, Andrew Morikoff and Maria Bisignano for their help with obtaining patient samples. And because we sent our geomics and cosmics to TAP, a big thanks to the NanoString team for performing all of these analyses for us. And thank you very much for your time.